Hello, and welcome everyone to Bio-Rad's webinar series on Droplet Digital PCR. This is our first webinar in a series of 6 Bio-Rad will be hosting on ddPCR. My name is Mohini Patil, and I'm the Global Product Manager for Droplet Digital PCR at Bio-Rad, and your host for today's webinar. First, let's discuss the webinar logistics. The webinar will last approximately 1 hour. Dr. Karlin-Neumann will present an introduction to ddPCR and its applications for about 45 minutes. Following the presentation, we will have about 15 minutes of live questions and answers with George. Throughout the webinar, feel free to use the Q&A box on the left-hand side of your screen in order to submit your ddPCR questions directly to us. We will get to as many of your questions as we have time for. However, if we cannot get to your question today, we will personally follow up with you over the next few weeks. Also, if you have any technical difficulties during the webinar, please use the Q&A box to report them. Now, a little bit about our speaker, Dr. George Karlin-Neumann. George received his PhD in molecular genetics in 1990 from UCLA. Following his postdoctoral studies with Dr. Ronald Davis at Stanford University, George co-founded ParAllele BioScience in 2001, which was later acquired by Affymetrix in 2005. George is currently Director of Scientific Affairs at Bio-Rad's Digital Biology Center, where he is responsible for establishing and overseeing collaborations. The title of his talk this morning is The Power of Partitioning, Droplet Digital PCR and its Applications. Without further ado, let's get started. George?
Thanks, Mohini, and welcome to everyone who is joining us today to learn about some of the many remarkable ways in which Droplet Digital PCR can enhance your applications. What I'd like to do is to start by describing what is digital PCR to those who are not yet familiar with it, and then I'll proceed to describe how Droplet Digital PCR, or as I'll refer to it, ddPCR, is performed on Bio-Rad's QX100 and recently launched QX200 Droplet Digital PCR systems. And then I'll follow that with a review of some performance data for the QX100 and 200 systems, interleaved with some published customer data. And that will cover areas of absolute quantitation, copy number variation, rare event detection, and finally a little bit on next-gen sequencing result validation. So a couple of the thoughts I hope you'll take away with you today is, one, to get an appreciation of how partitioning enables molecular counting. Secondly, that Bio-Rad's QX100 and QX200 systems are very versatile molecular counting instruments. And finally, that both of these systems enable very high precision measurements, very high sensitivity, reproducible, and enable scalability for a wide range of applications, as you'll see, and diverse sample types, including the capability for doing high throughput testing, and I'll illustrate each of these. Now, I want to step back for a moment and just consider with you how it is that we, as scientists and as biologists, investigate questions of interest. And you'll note that we typically look at a system under study and we identify various components of it, and often then we want to count them and to try and understand how the number of the component under question, how that actually varies under different conditions. On the molecular level, this is found often in, say, gene expression studies where we want to enumerate changes in transcripts under different conditions. For infectious disease, we may often be interested to understand what sort of pathogen is involved and what levels that pathogen are present, viruses, bacteria, and so forth, different stages of disease. In studying cancer, we're interested potentially in identifying what mutations or changes in copy number are occurring. In the case of next-gen sequencing, we oftentimes want to quantify very carefully how much of our library we're actually placing onto a sequencing cell. Looking at environmental issues, there's a common need to be able to assess whether or not there are contaminations in foodstuffs, such as for GMOs, and many, many other applications. And what we find is oftentimes our ability to understand further a particular biological process is limited by the precision, the sensitivity, or the reproducibility of the measurements we can make. So to put droplet digital PCR in perspective, I think you're all familiar with the power of polymerase chain reaction of PCR to be able to identify, detect the presence of as little as a single molecule present in a sample. If you look at the slide on the left, you can see sort of some of the earliest means we had of actually detecting the success of a PCR reaction. And you see that there was done in sort of a qualitative fashion to run a product on a gel and to see a desired band to identify that we actually had detected the sequence of interest. As the technology advanced in the middle 90s, real-time PCR, also called qPCR, became a favorite means of deploying polymerase chain reaction. There, oftentimes people are satisfied with making relative measurements relative to a reference gene of interest. However, quantitation is possible, although it's through the means of a standard curve, and there's some need to associate the experimental sample value with that of the standard curve, and the indirectness of it allows for noise and greater uncertainty in the measurements. Now, what we're referring to as sort of a third generation of PCR, digital PCR, is one in which we actually no longer rely upon a standard curve. And as you'll see, we manage to run reactions to endpoint in such a fashion that we get greater sensitivity and are less sensitive to inhibitors and various other conditions that can introduce noise into the PCR measurement. So let me go into about how it is that digital PCR actually operates. So in this first cartoon, I'm showing you a sample, and you can think of that as a typical PCR reaction sample, but rather than running it in a single unpartitioned volume, say a 20 microliter volume, we're instead going to divide that sample into many, many, many small compartments or partitions. In the case of the QX100 and 200 systems, every reaction is divided into 20,000 droplets of uniform size. So each of those partitions is a little microreaction, and we place the reaction in a normal thermocycler and run endpoint PCR on it. At that point, a droplet either has a target of interest in it and has a signal as shown by the three orange circles there, or it lacks it. And basically, you convert the presence or absence of a target in each droplet into a 0 or a 1, a positive or a negative. In contrast, and from this, we basically determine the concentration of the sample, of the target of interest in the sample. Now, we can operate this under a couple of different conditions. At low target concentration, which is sort of the classical condition under which digital PCR has been practiced, the number of target molecules is much smaller than the total number of partitions, shown here by only 3 orange droplets amongst many blue or 0 negative droplets that don't have the target. So partitions have either 0 or 1 targets in it. We count the targets, the positive droplets, and essentially that count tells us how many target copies of interest are present in the volume of sample that has been queried here. So it's very straightforward. If we look, however, at another situation that I'm going to emphasize in a little bit, we have high target concentration, and here you can see that a much larger fraction of the droplets are shown as positive, and furthermore, that some droplets have more than 1 target in them, 2 or 3 or even more, as I'll show you a little later. And in this case, you may wonder, how do you count the target molecules? Because you're converting each droplet, each positive droplet, whether it has 1 starting copy of your target or more, you're converting that into the same final signal, same final amplitude fluorescence. And how we do that is by using the Poisson statistics, which are described briefly in the lower right of the slide. And here we use the well-known Poisson statistic treatment to infer from the concentration of positive droplets and the number of negative droplets, what original concentration solution that combination of positive and negative counts was derived from. And so the equation is simply given by the negative log of, in the parentheses, essentially the fraction of negative droplets, 1 minus the fraction of P or positive droplets, or as I said, the fraction of negative droplets. And you'll see some of the ramifications of that, but I just want you to note that even when you have one target copy in every droplet, because of the random distribution of targets into these droplets, you'll actually still have 37% of your droplets are negative. And that's critical because in order to be able to make an estimation of concentration in digital PCR, you need to have at least some number of negative droplets and some number of positive droplets. And we'll go into that a little bit more in a subsequent slide. Just to start off to give you some sense of what are the benefits that you can anticipate from deploying droplet digital PCR is, as I said earlier, you get absolute quantitation, so you have no need for a standard curve, and some of the difficulties that engenders in actually having standards, reliable standards that behave the same way as your experimental sample, that may have matrix components that can alter the rate at which your fluorescence rises in a real-time reaction and lead to uncertainty there. You have greater precision that, as you'll see, replicates are very similar one to the next in the measurements they make on a given sample. And an important feature is also reproducibility. That's both experiment to experiment, day to day within a single lab, or also for labs scattered across the globe, that different researchers can get the same result reproducibly. Sensitivity is also a key measure, and you'll see the way in which precision, greater precision leads to greater sensitivity, where a measurement that has X amount of your target or half that amount, that you can discriminate those from each other more readily when you have very little uncertainty about each of those two measurements. And lastly, accuracy is something that you can also derive from digital PCR, but I want to emphasize that one needs to actually validate beforehand that your very, very reproducible or precise answer is actually giving you close to truth. Okay, so I want to move on now to giving you a little bit of an overview of what the Bio-Rad's QX200 droplet digital system looks like in terms of components. And this is essentially very similar to what the previously launched QX100 system looks like, but has some improved features. At the top here are the two instruments. There's a droplet generator, the smaller instrument to the right, and a droplet reader to the left of that. There's biochemical reagents on the lower left. There are oils that are used both for generating the droplets, and I'll explain about that more in a moment. There's also droplet master mixes that work together with the droplets to form highly uniform, well-performing PCR reactions in the droplets. There's reader oil in the larger bottles that's necessary to flow those droplets through the droplet reader. And to the lower right, there's a consumable, a disposable consumable that's used to actually generate the droplets from multiple samples in parallel very rapidly. So I'm going to move on now to the next slide and show you at a very high level what the droplet digital PCR workflow looks like. And then in subsequent slides, I'll expand on that a little bit to give you more of a hands-on feel for what the practice of the process looks like. So on the left side, again, you can see that there's the droplet generator, that again with the droplet generation cartridge I showed you a moment before, that droplets are generated from oil and sample, and this little crosshairs microfluidics within the chip. After forming these droplets, the reactions, the emulsified samples are transferred into a normal 96-well PCR plate that can go into a thermocycler shown in the second bubble. After normal PCR cycling, that then goes into, the plate is transferred into a droplet reader where the droplets are read as negatives or positives in two fluorescent channels as indicated there. And then lastly, there's a sophisticated suite of analysis software that turns those fluorescent amplitude signals into target copies, concentrations, or ratios as needed. So let me run through this now at a more, little more zoomed in view. And first of all, you can think of and trying to map this to your perhaps existing use of perhaps real-time PCR. You would make up a reaction in the same fashion as you would for real-time PCR. You would mix together primers and probes. For now, I'm going to be talking about TaqMan hydrolysis probe assays. You mix those together with one of various supermixes made by Bio-Rad to support either measurement of DNA, or additionally, there's one step, a reverse transcriptase ddPCR master mixes, and mix that as well with your sample. And then the next step that's different from what you normally do with a real-time reaction is to actually take your sample and to deposit that into one of these disposable cartridges, single-use cartridges that have 8 sets of 3 wells. One set is encircled here in the black, and you'll see that you add sample into each of the middle 8 wells with a multi-channel pipetter, your first 8 samples. You then add to that oil in the next set of rows. And finally, after this is placed in the droplet generator, droplets emerge in the 3rd well of each set of 8. Once you've placed your sample inside the DG cartridge, that then is secured on top with a gasket. Gasket shown in the middle. Need to go back one slide. Gasket shown in the middle here, and the entire assembly is placed in the droplet generator at the press of a button. Within 2 minutes, 20,000 droplets are generated in each of the 8 samples. Those droplets, as I mentioned earlier, are then transferred into a 96-well PCR plate, again with a multi-channel pipetter. At that point, the plate can be sealed and put into a thermocycler for a 2-step cycling routine, or one can continue to build up to as many as 96 samples in the plate, 8 samples at a time. And as I said, one heat sealable foil on top of that plate and places it in a thermocycler. After running a 2-step reaction, takes about an hour and a half to 2 hours. The plate is then placed in the droplet reader, which is essentially a droplet flow cytometer. It auto-samples each well one at a time, draws up the sample, and then runs the reacted droplets past single file past a detector shown in the lower part of the slide, and you can see the droplets going single file past a 2-color detector where either a FAM signal is detected in a droplet and or a hex or a VIC signal is detected. The next slide shows you, for a single well, one view, what we call a 1D or a temporal view of the data coming of the software, the QuantaSoft software program, which I'll show you a bit more about in a moment. The higher amplitude, this is shown for, say, a FAM, for example. You can see on the left axis, you have a fluorescence amplitude. You can see the upper band of blue droplets are your positive droplets, and the lower band at about 2,000 in this case, are shown in black, are your negative droplets. And so that represents roughly, of your 20,000 droplets, it represents your fraction of positive and negative droplets, as I said, is used to calculate concentration. You'll see in the next slide, again, this 1D or temporal view, again showing in the blue the same well I showed you for FAM, and you can have a view that also shows you for your second channel if you're doing duplex or higher plex reactions. You can see both the positive and negatives for those. Additionally, besides the 1D plots, you can also observe this very fruitfully in a 2D space, very much like you would do for FACS flow sorting, looking at populations. Here you have a very canonical cluster of view of 4 clusters. In the lower left are your double negatives. That there was neither target found in those droplets. In the upper left, you have the FAM-only targets. In the lower right, you have the hex or VIC-only targets, and then in the upper right corner, you have double positive droplets that actually have both FAM and VIC in them, and those double positive droplets get scored as the presence of both targets, of course. So there's a rich suite of software behind that, many other sorts of charts that can give you concentrations per sample, that can give you copy number measurements or fractional abundances of mutations and so forth, as you'll see as we go through the presentation further. I want to leave you with one other important point in understanding the droplet digital PCR system Bio-Rad has has made available, and that is that we operate under a different loading paradigm than people who are familiar with classical digital PCR are familiar with. And in the more classical paradigm, as I mentioned, people work under lower concentration. That's more or less shown by sample 2 in the slide, and I've just got a field here of like 143 droplets. And note that if you have a very low number of targets, such as 6 is what's shown here, actually, if you use the Poisson correction, it says you're really 6.2 is the concentration that gave rise to that number of positive and negative droplets. As you go to the next higher concentration, and that's about 5% filling of the droplets. When people operate in a low concentration, they typically are operating at maybe less than about 10% of the droplets being positive. So we move up to the medium concentration, you can see that in that field of there are 34 positive droplets among 143 shown. And when you correct that by Poisson, you get a slightly greater difference between the true concentration that gave rise to that. In this case, it says 38 versus 34. As you go to still higher concentration, which represents about 50% of the droplets being occupied, it says there are 70 positives there out of 143 total, but in fact, that really came from 96 targets because some of those droplets have more than one positive target of interest in them. And as I said, the Poisson correction is able to correct for what is the true concentration of that. This will become maybe a little bit clearer graphically what I'm saying, that even though we have 20,000 droplets and you need to still retain some negative droplets to be able to make a proper concentration call, that you actually can load into these droplets far more than even an average of 1 copy per droplet. You can go up to as much as 5 copies per droplet. And that's shown in this graph here, where on the bottom axis you have the fraction of positive droplets going from 0 to 100% of the 20,000 droplets. And you notice here I'm looking at what happens on the right axis when you have 20,000 droplets. The red line points to that. And at the same time, that represents an average of 1 copy per droplet of the target of interest. And you see again that on the lower axis there, that corresponds to about 63% of the droplets being positive, meaning there's still a significant number of negative droplets. Furthermore, you can see that this goes up well to, following the curve up, that even at 100,000 copies per 20,000 droplets of the target of interest, you have, or about 5 copies per droplet on average, you still have about 1% of your droplets being negative, and it's more than adequate to make a good concentration measurement, and I will show you that in some actual data in a moment. So again, this is a loading paradigm that gives us not 3 logs or so of dynamic range as you would expect if you were limited to having no more than 1 copy per droplet, but we actually can extend that by another couple of logs, giving us about 5 logs of dynamic range. So I want to shift now and go on to giving you some appreciation of the quality of the quantitation from this system. This first bit of data shows you the reproducibility and the linearity across the upper about 4 logs of concentration, and again appreciating that we can go well above the concentration of that you would otherwise get because of the incredible uniformity of the droplets that's shown here in the slide, the size uniformity that is. So in the first case, you can see that if you have 1 copy per microliter per about 1,000 droplets, you can see on the blowout there that you have a few positive droplets of the higher amplitude on the right side of the screen, and you have mostly negative droplets. As you increase the concentration by 10-fold, you have more positive droplets, high amplitude, and fewer negatives. As you go still 10-fold higher, you get even more positives and even more. And finally, at that's over a copy per droplet we're looking at there. As you get towards 10 copies per droplet, as I mentioned, you've converted almost every droplet to a positive droplet, although there are still about 3 remaining ones beneath the red line that you can see there. So that's pushing it a bit further than we like to. We recommend normally not going beyond about 5 copies per droplet where there are several hundred negative droplets still available. And what's striking here is that you see that it's basically a perfect R-squared across these 4 logs. And what you may not appreciate is that there are actually confidence intervals, 95% confidence intervals on each of those markers, and this was done on 3 different instruments. The markers are superimposed from the measurements made on 3 different systems, and they're completely superimposable. That's sort of an older view of the data. A newer view that's representative of what you would see in the QuantaSoft software, one of the many charts I referred to earlier, shows an experiment where you're looking at a 2-fold titration series of Staph aureus DNA that is spiked into a master mix sample that has 1,000 copies of the human genome per microliter, or essentially 1 copy of human genome per droplet, and we're scoring it here for the RPV30 gene, which you can see in the green at about the 1,000 concentration going all the way across. The blue represent markers for the FAM channel, which is where the Staph aureus is being measured, and you can see as you go up each of these 4 replicate series here, a single data point for each one of those, a single ddPCR for each of those blue dots. You can see that there's a very, very regular doubling of the counts for a doubling of the concentration. You can go down about another log from this, measuring really a few copies per well, and still those being legitimate positives as long as you don't have contamination in your lab. So here we're showing the dynamic range and the precision over a wide range. I want to zoom into a 2-fold range there and show you a little bit of how much higher precision can be obtained. Here you're looking at a zoom-in of that 2-fold difference in concentration, and here each of the blue data points has an increase of 10% from one to the next concentration in the series. And what you can perhaps see, I hope on your screen, is that on each of those markers there are a couple of little end caps to 95% confidence intervals that actually don't even extend beyond the size of the marker, which means that with 95% confidence, your true concentration falls within the concentration shown by that blue marker. And you can see that if those confidence intervals do not overlap with the next adjacent one, that you can distinguish those 2 measurements, and they represent probably 2 distinct concentrations with high certainty. So you can see here that you can make very, very high precision measurements. Now, moving on to the use of this in the real world and in customers' hands, this is a recent publication from Manish Tiwari's lab, where he was deploying the system for absolute quantitation of cancer biomarkers in serum and in plasma. One of the problems that he's faced is using real-time PCR to identify these biomarkers and to actually associate their relevance with the presence of a particular cancer or not in controls. They found that the very low quantities of these biomarkers made real-time an unreliable tool for using here. So they wanted to actually do a comparison to find out, did they actually get a more reliable and reproducible estimation of microRNA markers in the blood. They did a comparison here between, on the next slide, a comparison between real-time PCR shown on the right, on the top, and digital PCR on the left. In this case, in the upper panels, you're looking at one of 6 microRNAs they studied, MIR141, and this dilution series goes on the left from high concentration down to low concentration, and at the low concentration, you're detecting really just a few molecules per sample, and you can see on the droplet digital PCR results on the left that there is much less variance around the concentration values shown. And I will emphasize that in each of these 2 curves here with the qPCR on the right, which shows much greater scatter for each dilution point in the series, that actually they were testing for variability in measurement both within the same day, doing 3 different reverse transcriptase reactions, cDNA synthesis reactions on a dilution series of a synthesis target. They were doing that 3 different RT reactions. On top of that, they did 3 technical replicates of both digital PCR on the left and qPCR on the right, and they also did that same experiment over 3 different days, and all of that data for each of those data points is compressed into these 2 figures, and you can see the much greater uncertainty in concentration values given by qPCR on the right. After satisfying themselves that they had much greater data reproducibility, about a 7-fold increase, and a significant decrease in their CVs from about 37 to 86% with digital PCR. They applied this to some cancer samples shown in the lower part of the panel. These are prostate cancer samples where they have patient serum here. They're looking at cases and controls. And you can see on the left side of the graph that you've run these reactions both in digital PCR and you've run them on the left and on the right in real time. And the green circle shows the digital PCR a little bit off center there, but you can see between the cases and controls that there's a better discrimination there than there is for qPCR on the right. So it actually turns out when you look statistically at whether or not you can distinguish cases and controls based on that particular marker, you have significantly higher sensitivity and specificity using the droplet digital PCR. And it's only with droplet digital PCR that you can actually establish statistically significant association of that marker with prostate cancer in these samples. This is one of many instances in which the technology has been applied by users in the field to cancer applications, and I'm not going to say more about it in this presentation, but a month from now there will be another presentation by a colleague that will focus specifically on cancer. So I'm going to move on now to the usage of the QX100 and 200 in looking at structural variants. Specifically here I'm going to focus on copy number variations. And as you're probably aware, in the last 10 years or so we've become increasingly aware of the large fraction of the human genome which has appears to have amplifications and deletions occurring across the genome. There's roughly 10% or so of the genome that has copy number variations larger than 1 kilobase, some of these running into the hundreds and millions of base pairs, and there's nearly 30% of the genome that has amplifications or deletions at 100 base pairs and above. What that means biologically is something we're just beginning to investigate, and I'll say a little bit more about that in a few minutes. First off, I want to show you what the data looks like when you measure not a single concentration of your target of interest, asking what is the copy number of that target in a diploid cell. You're going to compare that to a reference gene that's a single copy gene. And here we're looking at MRGPRX, the nociceptor gene, and asking the question in a series of HapMap samples shown on the left, which are known to have certain copy number levels of the MRGPRX1 gene. We see samples that have copy 1, copy 2, 3, 4, 5, and 6. And you can see that in for each sample there are 8 replicates there. Each one of those represents a single replicate of that same sample, and you can hopefully see that the numbers are extremely reproducible from well to well, that a single measurement is sufficient to distinguish 1 from 2, 3 from 4, and so forth, and even 5 from 6. And that's something that is extremely difficult, if virtually impossible to do with a real-time system that has much greater noise, in which even with doing a number of replicates, it's difficult to get more than perhaps a 50% difference in concentration and thus copy number. Another powerful thing that you can do with the droplet digital PCR is you can actually merge data points from individual wells if you want to get still higher precision. And the data on the left is regraphed on the right after merging those wells together. And here you can see, just looking at 5 and 6 copy number samples, that the error bars, again, around the 5 and the 6 have shrunk down to almost invisibility there. So you have extremely high confidence that a sample that you're measuring as 5 is distinct from one that's measured at 6. And this can be applied to the system under study. So that's one way in which you can increase precision to the extent that you need it. Another way is shown in this slide, where we're comparing different inputs amounts. Again, looking at a number of HapMap samples across the x-axis there, about 20 HapMap samples of varying copy numbers, again for the same MRGPRX1 gene. And the ones that I've encircled there are just to draw your attention to distinguishing copy number 4 from copy number 5. 2 samples at 4, 1 at 5 in the middle. You can see again that the 4 replicate experiments run on 2 different systems that you can distinguish 4 from 5 with a single sample there. And note that when I increase the input DNA by tenfold up to about 66 nanograms, which turns out to be about 1 copy per sample or 20,000 copies per well, that now the confidence intervals are much smaller. The values are still the same, but you have greater confidence in them, and you can distinguish even more clearly 4 from 5. So the system is very tunable, if you will, to being able to generate the degree of precision that you have depending on the kind of sample you have available and the small differences you're trying to distinguish one from the other. And also to note that across 4 different runs of this experiment shown by the 4 bars in each of the samples, that it's highly reproducible. You get the same value, very similar confidence interval when you run the experiment multiple times. So just to put a point on that, again, you can extend this to higher number copy number samples. In this case, it's showing a CCL3L1 gene, a series of copy number states in different HapMap samples. You can distinguish nicely copy number 10 from copy number 11. And by extension, we've had users that have actually been exploring interesting copy number genes that extend up in the hundreds. So with sufficient amount of sample, one can actually discriminate very large copy number changes. One other point that I want you to take away is in this next slide is the ability to really do high throughput analyses with the QX100 and 200 systems. As I said, you can run 96 samples per plate. You can run a couple of assays per well. Here we're doing target and reference where the target is a CYP2D6, and we're comparing that to, among other reference genes, the RNase P gene or RPP30. And here we've taken a series of HapMap samples from the 1,000 Genomes Project, either British or Kenyan samples. And you can see that in the British samples up in the left, for example, that when we've rank ordered the samples, the ones that are copy number state 1, copy number state 2, or copy state number 3 are easily distinguishable one from the other with a single ddPCR well per sample. So this is three plates of samples that have been analyzed by a single person in a single day in a normal 8-hour day, that this is the kind of throughput that can be achieved. And where might that be of interest? Well, the next couple of slides show you the deployment of this technology in the hands of workers in Steve McCarroll's lab. This is a work published by Linda Butker and et al., trying to understand interesting structural variation in chromosome 17, where they have a 1 megabase inversion where certain medically interesting important phenotypes map for both neurological disease and female fertility. And it's a complex region that has both, as I said, an inversion, and within that region there are multiple repeats, such that different individuals have different copy number states for each of those repeats. And I'm going to show you that in the next slide. So what they've done is looked at several hundreds of samples from the 1,000 Genomes Project and very rigorously assessed copy number states of various regions within that. And this next slide shows you in the upper part a little map of that region. And within that megabase, as I said, there are 3 regions for which they've made TaqMan assays that can actually enumerate that in Droplet Digital PCR. And in the second row, B through D, you can see a histogram of copy number states or copy number alleles at these multi-allelic loci that show for region 1 in B, region 2 in C, and region 3 in D, show the frequency of each of those alleles as assessed by next-generation sequencing. In the next row beneath that, the histograms show this similar data for a partially overlapping set of about 100 samples in Droplet Digital PCR, and those same allelic states are present. When you look in the bottom row, H, I, and J, at regions 1, 2, and 3 respectively, and you look at the correlation between the data from next-generation sequencing to Droplet Digital PCR, you can see that the correlation is nearly perfect. Concordance is greater than 99%. And in fact, in often cases, you can see there's greater dispersion in the next generation sequencing results shown on the x-axis versus the droplet digital PCR axis on the y-axis. So this capability of getting rapid and very precise copy number measurements has been extremely valuable to the McCarroll lab in being able to rapidly score populations for multi-allelic loci that occur not just in this interval, but actually across the entire human genome with many biologically important loci, some known and some to be discovered there. So this will remain a very important tool for discovery and association of copy number alleles. I want to move on now to another use of the system, and that is what we refer to under the umbrella term of rare event detection. And this is actually referring to a couple of different types of sensitivity needs. The first of these is shown in the upper, and we refer to that as rare sequence detection, where you have a rare sequence such as, say, a viral sequence or a GMO transgene that you're trying to detect in a background of very dissimilar DNA. So it could be like an HIV sequence, which I'll show you in a moment, against the human background DNA. And in that case, you can measure with a single assay, a singleplex assay, the target of interest. And if you choose to, you can ignore measuring the background assay, which enables you to load very high amounts of DNA, a microgram, microgram and a half, as to say hundreds of thousands of copies to 500,000 copies of your background DNA, and say, is there a needle in that haystack? The first example I'm going to show you is of that. Subsequently, I'll go on to actually looking at rare mutations, which is oftentimes very powerful in looking at studying cancer etiology and treatment, and that in this case we use a couple of assays directed simultaneously either against the wild type allele or the mutant allele, and I'll show you that in a moment. First off, though, we're looking at the rare sequence detection, and just to see what the system is capable of, this is an experiment done by Wei Yang, which is showing you the ability to detect one HHV6, human herpes virus 6 genome in about 1.7 million background copies of human DNA. And just to kind of walk you through this quickly from the left side, moving towards the right, I can show you first on the left there are controls where there's a modest amount of the HHV6 DNA that was spiked into a Raji human DNA background. And you can see where the controls are, the amplitude of the expected positive droplets detecting the HHV6 are shown relative to the negative droplets that are shown below 500 in terms of the amplitude there on the bottom. If you move to the next set of wells to the right, labeled NTCs, this is where you have the same assay being run in the same similar droplets, but where you've put in neither human background DNA nor the HHV6 DNA, and you see there are no positive droplets coming up. So it's very clean. If you move on to the next 60 wells that say negative controls, in each of those there's no HHV6 DNA, but there are 25 genome equivalents, or about a microgram and a half of DNA. That is per droplet 25 genomes, or about a microgram and a half of DNA, nearly 500,000 human genome copies of Raji DNA in each of those 60 wells. And then you say, do I detect any false positives in the HHV6 DNA? And you see again at the expected amplitude around 4,000 or so, there are no positive droplets. So the false positive rate was zero, basically measured here. If you then look at the kind of the experiment to the right and say, if I now put in an extremely low amount of HHV6 DNA at a concentration where you expect to have about 4.5 positive droplets, HHV6 positive droplets in 8 million human genomes, you in fact get 4 positive droplets. We were extremely lucky to get so close to the expected, but the fact is that they're bona fide positives, and you have a sensitivity that's well beyond 1 in a million if you have very clean practices for PCR and clean solutions. So one very powerful realm in which this has been deployed recently is in the HIV detection community. And investigators that have been working at trying to eradicate HIV recognize that that we have very effective antiretroviral therapies that can knock down the viral load to perhaps undetectable levels, but there are still reservoirs of latent virus, provirus, that's integrated into the human chromosomal DNA, and this DNA is a source potentially of a reemergence of the infection. So people have struggled to try and make the most sensitive measurements possible to determine if a patient is really totally free of the virus or not. And in these efforts, they are always looking for means by which they can more sensibly detect the presence. And Doug Richmond's lab down at UCSD, led by Matt Strain in this effort here, has published on the comparison of droplet digital PCR for sensitivity relative to qPCR, and he's found that they can go down probably 10-fold to 100-fold more sensitively with droplet digital, in part because they're able to merge together many wells to be able to actually challenge, as shown in this next slide, as many as about 10 million copies of the human genome, and to screen through those in numerous wells. So this is just showing two of the targets that are used, are detected for looking at viral latency, both the Pol gene in the provirus and in the 2LTR episomes in the nucleus, as well as just in the 2LTR itself. And the hatched region shown in the lower left is a region which they can make discriminating measurements with droplet digital PCR, but they cannot with real-time PCR. So these assays have been adopted within the HIV community, and we're starting to see a number of efforts being conducted and published, including this one in this next slide, where it was deployed by a study from Dr. Deborah Presseau at Johns Hopkins in trying to assess whether an infant that was born HIV positive, treated immediately after birth with intensive antiretroviral therapy, then was serendipitously off of treatment for about 6 months, and when the infant came back and they tested for virus using the droplet digital PCR system, they could find no detectable virus left in the infant, the toddler at this point. So it was deemed the second functionally cured patient with antiretroviral therapy, and has sparked a lot of interest in the field for what are the possibilities for preventing infection in newborns, as well as the potentials for trying to deploy that in adults. This has also been used to assess the now famed Berlin patient who was also apparently cured of HIV during a bone marrow transplantation, and so forth. So it's been extremely valuable in the HIV detection community and beyond. Now for the second type of droplet digital PCR, we have here the case where you're looking at a rare mutation, and partitioning enables you to actually take the ratio, the very imbalanced ratio. You normally have a wild type assay and a mutant assay, and has that makes it much more possible to detect the mutant DNA against the background, where normally the incomplete specificity of the assay is obscured in a non-partition reaction. So on the next slide, you can see this in a BRAF, where I've got a titration and going from basically 1% mutant abundance all the way down to about 1 in 100,000 abundance. This is also visible in other assays activating mutations such as KRAS G12V, where mutant only is shown in the upper left, the absence of mutant is shown in the right, and as low as 0.001% is shown in the middle, and a single mutant is detected there, and several 100,000 copies of wild type, and similarly in between. I'm going to have to kind of speed through the last of this to make sure that we have time for your questions. So I'm just going to refer you to the fact that not only in cancer, but this is also being used now in transplantation monitoring of patients, which is a very expensive procedure, and which is very critical to assess the success of the transplant, to monitor the patients regularly. It's found that droplet digital PCR can be used to both monitor the levels of donor DNA in the blood, and to assess whether or not those levels are rising above undesirable levels in either kidney, heart, or lung transplants. Here on the graph on the right, you can see the change in the donor DNA copies in the blood, liquid biopsy as done in cancer, going from very high levels shortly after the transplant in the upper graph down to very low levels, and then monitoring in the lower panel across many months of treatment. So this can be used to try and correct for excessive immunosuppression, use of immunosuppressive drugs, and to respond more quickly to potential rejection incidents and to potentially save an otherwise organ that may be rejected. I'm going to just quickly run over another example of the use of NGS validation for the technology, and this is an example that I'll let you look at in more detail yourself, in which droplet digital PCR was used by the Snyder lab to confirm or to challenge results in both heteroallelic gene expression, difference in the expression of the two alleles during the course of a viremia, as well as to look at RNA editing to confirm those results. And that's shown in the next slide, but I'm going to let you go through that subsequently if you wish to download the slides, and once again confirms the greater precision and certainty that can be found using the droplet digital PCR measurements. So lastly here, I just want to point out that this has now been in the hands of many investigators for several years now, and there's an increasing stream of peer-reviewed publications using it. There's now nearly 50 that have accumulated to attest to its widespread applicability. The last thing I want to just leave you with, and just take about 2 minutes for that, is just to say that with the launch of the QX200 system several months back, now we can go beyond using just TaqMan hydrolysis probes. There's the ability to use a DNA binding dye, EvaGreen, requires no TaqMan probes, just well-designed primers. This gives very, very high performance, and shown briefly in this next slide, we have show low abundance and high abundance genes being detected over 4 logs of range with extremely linear detection. And I want to leave with sort of a last point about the use of the EvaGreen dye, is that it's actually enabled us to see some things that were not readily available earlier when we were using TaqMan probes. This slide shows you how we were able to actually detect simultaneously and quantitate immature unspliced RNAs, which we refer to as a supercluster in the panel on the left, at the same time as we're able to quantitate the lower amplitude predominant mature RNA due to differences in the size of those amplicons, and thus differences in the dye binding, resulting in differences in the amplitude. So at the same time as you get digital information with the droplet digital PCR, you get some analog information which can give you some extremely interesting insights, and that's shown with 3 other genes on the right as well. So it's a common phenomenon that enables you to look more deeply into the biology of your system. And last point is to say that it's also enabled us to look at some instances in the case of telomerase activity where we can actually measure the activity of the enzyme on a DNA substrate that is very repetitive and is not readily amenable to design of TaqMan probes. We have, in conjunction with Elizabeth Blackburn's lab at UCSF, we have helped them to convert what was previously a radioactive trap assay into a droplet digital PCR assay, shown in the next slide. And I won't say more about this in this talk, but again refer you to a colleague's talk next month, which will focus in on cancer applications. So lastly, I just want to say there's a wide range of applications that can use that can deploy the droplet digital PCR with EvaGreen. And just to finish off here, I hope you have been impressed that the QX100 and 200 systems are highly precise, sensitive, and reproducible molecular DNA counters, that they do not require a standard curve for quantitation. The precision and sensitivity is something that's tunable by the number of wells you use, from 1 to as many as you need. That it works with a wide range of sample types. It's easy and versatile and affordable, and it works with or without TaqMan hydrolysis probes. So lastly, just want to thank all the very capable colleagues who have helped to develop the system and to develop its use in various applications. Thank you, and that's it. Thanks for your attention.
Thank you, George, for that very engaging talk. For everybody attending today, if you would like a copy of today's PowerPoint presentation or a copy of the QX200 Droplet Digital PCR brochure, they're available at the link to the left of your screen for download. The webinar is also available on demand within 24 hours. If you would like any additional information, please feel free to reach out to your local Bio-Rad Droplet Digital PCR specialist as well. George will now take questions. So the first question we have today is regarding the reaction volume. So what is the reaction volume for a single ddPCR run?
Okay, thank you, Mohini. The volume for reaction is 20 microliters that is emulsified in each sample well. Plain and simple.
Okay, so the next question is moving towards an assay. Can droplet digital PCR be used to evaluate the number of targets with a particular microsatellite repeat within a certain locus?
It could conceivably be used for that. That depends upon knowing, having an assay that's specific for the target of interest, and then being able to have another assay, reference assay, that gives you a measure of number of genomes that you're scoring that in.
Okay. We have another question regarding assays. Can one use MGB TaqMan probes with the QX200?
Yes, most certainly. The QX200 not only allows you to use the same TaqMan hydrolysis probes as work in the QX100 system, but also adds on the ability to use the non-TaqMan EvaGreen dye binding detection. And this will work, the TaqMan hydrolysis probes work with both MGB probes as well as non-MGB.
Okay. And then another question regarding assays. Does Bio-Rad provide validated assays?
Good question, Mohini. I neglected to mention that. Yes, Bio-Rad has committed itself to providing a large repertoire of validated ddPCR assays, both for rare mutations such as activating mutations in cancer, and a growing panel of copy number assays also validated, moving up into the hundreds and growing quickly.
So now moving to a question regarding the number of targets that can be analyzed at one time. How many targets can be analyzed, and does multiplexing decrease sensitivity?
Okay, the answer on multiplexing is that yes, you can do multiplexing. Of course, I showed you for doing duplexing in the two color channels, but you can also multiplex higher level than that. I would say conceivably up to 4 is not inconvenient with the current system, and you do that through being able to take advantage of differences in amplitude of your clusters depending on the size of the amplicon. And as I mentioned for the EvaGreen, a larger amplicon gives you a higher fluorescence. Interestingly, it's the inverse of that for TaqMan probes. So with both systems, we've managed to multiplex by both a combination of the two dye channels combined with amplitude variation to position clusters in ways that you can distinguish as many as 4 targets in a single sample well. And as far as sensitivity is concerned, it should not diminish your sensitivity because it actually gets simpler at lower concentrations of the targets. It produces a simpler constellation of clusters. I will add that the software, it still requires some manual inventorying of the concentrations of your different targets, and that the software will soon catch up with that and make that an automated process.
Going back to the question regarding the assays, validated assays, do you have an EGFR validated assay?
The answer is yes, although as the listener probably appreciates, there are a number of assays that can be activating mutations for the EGFR gene. and we have a number of those available, yes. And that content should be, I think, available on the website to see what is the increasing inventory of cancer gene mutations and copy number assays.
And then we have a question regarding the instrumentation. Are there any plans to manufacture under ISO 13485 or QSR?
There most certainly is, and we do expect to have systems that will be built under those conditions, both for CE marking and ultimately for FDA approval.
Okay, and then one last question. How many technical replicates are recommended for droplet digital PCR, or is there a need for technical replicates when doing ddPCR?
That's a good question, and I would say that ultimately a single replicate is quite satisfactory and highly reliable. I think you may recall having seen the slide I showed on the copy number estimation of 1,000 genome sample HapMap samples, that you could do 96 different samples per plate, each at a single well, and get a very reliable estimate of the concentrations and copy number for each of those samples. I think when you're first starting, it's natural to want to do a couple of wells, a couple of duplicates, just to see that you're getting the same answer in each well. Ultimately, I think you can let go of those training wheels, and if your throughput demands it, to go with a single replicate per well, per sample rather, I meant.
Well, thank you, George, and thank you everyone for attending the first Bio-Rad Droplet Digital PCR webinar this morning. We have 5 more webinars in the coming months, so please participate in the full webinar series. As a reminder today's webinar will be also available on demand within the next 24 hours. For those of you who had questions we were not able to get to today, someone from Bio-Rad will follow up with you in the next few weeks. For more information on Droplet Digital PCR and educational resources, visit www.biorad.com/webinar. This URL is displayed to the left of the slide in the resources box. Thanks,